Differentiation, de-differentiation and redifferentiation. Differentiation is that process in which cells, when they divide from apical meristems of root and shoot regions as well as cambium, then they mature to perform specific functions. This whole process is known as differentiation, and when they mature to perform specific functions, they lose their capacity to divide again. But under certain conditions, they regain their capacity to divide, and hence this process is known as de-differentiation. Now these de-differentiated cells, they divide and again mature to perform specific functions, and when they again loses their capacity to divide, it is known as redifferentiation. Differentiation is, that is, they are coming from the meristems getting mature performing specific functions. Differentiation is happening. When they are performing, they are mature, they are performing specific functions, they cannot divide. But under several conditions, they will get the ability to divide again. That is, de-differentiation will occur. Once they have divided, they again will mature and perform various functions, but now they cannot divide further. Hence, the process is redifferentiation. Development. You can see in the diagram, it starts from meristematic cells, leads to plasmatic growth. Plasticity is that ability of plant in which, depending upon variable external environmental conditions, they grow different kind of parts to sustain in that environmental conditions, and it is known as plasticity. Meristematic cells shows plastic growth. Here the cell division you can see, it leads to differentiation. In plasmatic growth, expansion takes place. Differentiation leads to maturation. Mature cell leads to senescence and hence death. Plant growth regulators or PGRs. These are small simple substances or molecules of diverse chemical composition. They can be indole derivatives, they can be adenine derivatives, they can be terpenes, they can be gases, they can be keratinite derivatives. Discovery of plant growth regulator. Darwin, along with his son Francis Darwin, initially for the first time showed how plant growth regulators are working. It worked on the clupetile of canary grass and shows the function of auxins. Then later on, F. Scooch and 3 other independent researchers gave us the composition of various PGRs as well as plant growth inhibitors, commonly named as abscisic acid. Physiological effect of PGRs. Auxin. Auxin is chemically indole 3-acetic acid. Synthetically or humanly synthesized auxins are NAAN24D. They help in plant propagation as they help in promotion of development of roots on the stem. It helps in flowering in case of pineapple. It prevents the early drop of fruit and leaf, whereas it promotes the drop of senescent leaves or fruits. It induces parthenocarpy, and it is also used as weedicide or herbicide. 2,4-D is the commonly used weedicide.